Well, good evening, ladies and gentlemen. It is so good to see all of you here. My gosh, we've made it. To California, yeah. Fantastic. We are over 550 strong registered for the conference. Yeah. And an incredible program that I am so thrilled to kick off tonight. For those of you who I haven't had the opportunity to meet, I am Robert Riggs, and I have the great honor of being the CEO of the Scleroderma Foundation and one of your hosts for this weekend. And I just want to thank you all for mustering up the energy and the excitement to be here for what is going to be an incredible weekend. I can't start the weekend without thanking a couple of people. More than a couple, actually, but it's very especially Carrie Connolly, our Programs and Services Director, and the Conference Director. Chris Pettit, President, and Jerry Capel, Interim Executive Director, and the board and staff and volunteers of the Southern California chapter who are helping us so well this weekend. Bill Martin, President, and Kelly Davidson, Executive Director of the San Diego chapter, because many of the volunteers are coming from San Diego as well. And none of this is possible. It's a huge enterprise to undertake, and we cannot do it without some significant financial support. So I have to give a shout out to Actelion Pharmaceuticals, our national gold sponsor again this year, our silver sponsors, Bayer Healthcare and Gilead Sciences, and our corporate sponsors, United Therapeutics and Genentech, and all of our exhibitors who are in the next room, who I want to make sure you visit this weekend. They are very interested to meet you, to learn more about this disease, and to be a support to you. I want to thank our medical advisory board, who are the champions of the faculty that you will hear from this weekend. They do an incredible work in supporting us year after year, donating their time, taking time off from their clinics, helping their patients at home so they can be here and help all of us. But also, I want to thank you, because we are here for you. And all of this would mean nothing if there weren't all of your faces in these seats here tonight. You make it possible for us to do this type of programming. You inspire us to continue as an organization to do what we do and to find new and creative ways to make it better. And hopefully, for those of you who are first-time attendees, which is over 2/3 of you this weekend, you're in for a real treat. For those who are old-timers who have done it before, I think you're also going to be in for a treat, because thanks to my incredible, amazing staff in our office in suburban Boston, we put together a program that is beyond what you'd think we could do. And I think that you will see that we have 79 presentations, with 48 of them being offered for the first time. Yeah, a hugely expanded juvenile program this year. And I can't think of a better way of kicking off our weekend than with our opening keynote speaker. There are really no words to express the deep respect and admiration I have for Dr. Carol Feghali-Bostwick. She is really, quite literally, in a class by herself. A determined and successful scleroderma researcher, Carol further distinguished herself among her international colleagues with the promising research on the E4 peptide that has shown promise in not just stopping fibrosis in laboratory models, but also reversing it. Her cutting-edge research, published in the peer-reviewed medical journal Science Translational Medicine in 2012, was also called out as one of the most significant breakthroughs in scleroderma research over the past 2 years by her peers at the 3rd Systemic Sclerosis World Congress in Rome this past February. In addition to running a research lab and mentoring new generations of scleroderma researchers, Dr. Feghali-Bostwick is also the Kitty Trask Holt Endowed Chair and a Professor of Medicine in the Division of Rheumatology and Immunology at the Medical University of South Carolina in Charleston, a true powerhouse in scleroderma research and treatment. She is the Vice Chair of the Scleroderma Foundation's National Board of Directors, and she is the Chair of the board's Research Committee. She's a tireless volunteer for the cause, and in her free time, she manages the challenge of being a mom to 3.5-year-old twins, which, when measured against her exceptional and extraordinary accomplishments in her career, is probably her greatest source of pride. So please join me in welcoming the person that never says no when asked to do something to assist the Scleroderma Foundation, Carol Feghali-Bostwick.
Good evening, everybody. It's wonderful to be here. It's wonderful to be able to experience the conference with you, to join you, to start it off with you. But it's also wonderful to have a chance to talk with all of you and feel the energy that's flowing throughout the weekend. I'm thrilled to be here. I'm honored to be here, and I hope you will enjoy the evening. What we're going to do is start off with a talk. And I'll tell you where we are with our therapy to keep you posted on it. But what we're also going to do is have a quiz at the end. So you have to pay attention. And the quiz comes with prizes. So there's incentive. All right, so in order not to make it a late evening, I'll go ahead and start. As Robert mentioned, I am in Charleston, South Carolina. I relocated there about 9 months ago, and this is the bridge I cross every day on the way to work. It's called the Ravenel Bridge. And I want to talk about accidental discoveries or serendipity in research, because so many of the discoveries that affect us every day, things we use in everyday life, drugs we take, everything else happened accidentally. And I think you only appreciate how research happens when you hear about some of those examples. So I first want to start off by thanking my lab team who helped with all the research we did, especially a superstar at the top, Dr. Yuki Yamaguchi, who's rising through the ranks very quickly in Japan, in Yokohama, because of exceptional work she did on this peptide. I was very fortunate that my lab team moved with me when I relocated to Charleston, so we had very little downtime, so I'm very grateful for them. So research is important. Why is research important? From the perspective of a scientist, I'm a nerdy scientist, it's easy for me to say that, but from your perspective, why is it important? Because the discoveries that result from the research are the ones that lead to the therapies. A lot of the treatments that we use for a variety of diseases started with research that was in a bench research in a laboratory that ultimately led to a treatment. So how do we do research? We start by proposing a question. You pose a question, and then first thing we do is go read what's known about this topic that we're asking, because you want to make sure that what you do is something that hasn't been done before. You don't want to be reinventing the wheel. Once you read up on what's been done on the topic, you then formulate a hypothesis, and then you design experiments that will test your hypothesis. And then you do the experiments, you look at what results you get, you analyze them, you interpret them, you draw your conclusions, and then you share your findings in the scientific community so that other people will know this has been done, this is the information we have, and they can use that information to then guide their research. So that's how the field is advanced. This is an illustration of what a research lab looks like. I realize that a lot of you haven't been in one. You're welcome to come visit ours anytime you're in Charleston. We do a lot of tours of the lab. But this is a typical bench in a research lab with reagents, biohazard containers, equipment, everything else, and that's where a lot of the research we do happens. So we approach scleroderma as a puzzle. It's a research puzzle, and we're solving the puzzle, and the puzzle gets solved piece by piece. Many of you know that one of the problems in scleroderma is excessive production of collagen, and this collagen ends up laying around the cells, connecting and making the tissues thick. So thickening is another word for fibrosis. And so we look at the collagen, we look at other features of scleroderma, we look at different organs. Most of our research is on skin and lung right now, but there are people looking at kidney and heart and other organs. We grow cells called fibroblasts. These are the cells, when you've been asked to do a punch biopsy for research, those are the cells that we grow out of the punch biopsy to be able to do our experiments. In addition to looking at all the aspects of scleroderma, there's another reason why scleroderma is key to look at in all these organs. So if you notice from the puzzle pieces, scleroderma affects the lung, affects the kidney, affects the skin, affects other organs. So why is it important to study scleroderma? Because we consider it a prototypic disease. The hallmark of scleroderma is this fibrosis or thickening that happens in the skin and internal organs. But fibrosis is not unique to scleroderma. Actually, fibrosis that happens in different organs is characteristic of other diseases as well. And if we can solve the puzzle of fibrosis in one, whatever therapy we develop is likely to work in the others as well. And actually, if you look at the numbers, and this was a study done by someone who works at the National Institutes of Health, Thomas Wynn, had reported that almost half of all deaths in the developed world are because of fibrosis. That's a large number. That's a lot more than cancer. That's a lot more than a lot of other diseases. And the reason that fibrosis causes nearly half of deaths in the developed world is because fibrosis is a feature of many diseases. You'll recognize many of these. Liver fibrosis happens in alcoholic liver disease, hepatitis C, hepatitis B infections. It can be autoimmune, a lot of reasons. Heart fibrosis occurs. If you've had a heart attack, the scarring after that is fibrosis, in addition to a variety of diseases. Skin fibrosis occurs in scleroderma, but it occurs in other diseases as well that you've probably heard about, such as keloids. Kidney fibrosis also occurs, even in people who have diabetes, they get kidney fibrosis. Lung fibrosis is in scleroderma, but airway fibrosis in the lungs is in asthma. Idiopathic pulmonary fibrosis is a horrible disease that has a mortality of 50% at 2 years. That's very much like lung involvement in scleroderma. So you can see in the eyes you get fibrosis. So nearly every organ in the body can be affected by fibrosis depending on the disease we look at. That's why solving the puzzle in scleroderma will be key to solving the puzzle for a lot of these other diseases. So how do we do this? And I'm now going to guide you with an example of how we did our research. So we started out with a skin punch biopsy. Those of you who have had it know it's about the size of the back of the back of a pencil, so the eraser attached to a pencil, it's about that size. We get a punch like this one out of the skin, and we grow these cells. They're very elegant cells. They're spindle-shaped, and they grow on plastic, basically, in the lab. And out of a tiny 3-millimeter punch biopsy, we can grow millions and millions and millions of cells that help us for a lot of experiments. So what happened to us is we were looking at these proteins, and the proteins we were working on were called insulin-like growth factor binding protein, IGFBP. And we had found that those proteins were produced in excess in scleroderma patients. There was too much of these proteins in scleroderma patients' skin, in scleroderma patients' lungs. But we didn't know why. Were they there just as a consequence? Were they bystanders? Or were they involved in causing the fibrosis? And basically, we did a series of experiments that showed us that these proteins can cause fibrosis. If you add them to cells, normal cells, fibroblasts, in the lab, the fibroblasts will become scleroderma fibroblasts. They'll make more collagen. If you add these proteins in the skin or lungs of mice, they will get fibrosis or thickening of the skin and lungs. So we figured they must be involved early in the process, promoting the fibrosis that we see in scleroderma. The question was, how were they doing it? So learning how they do it, you can learn how to stop them from doing it. And around that time, we were in Normandy on vacation, and it was the 60th anniversary of D-Day, and there was an exhibit for the history of science and physics in one of the old castles in Caen in Normandy. And while looking at the exhibit, I came across a quote from this physicist that said, "I have never tried to dissuade a man from attempting an experiment. Even if he does not find what he is looking for, he may find something else." And the reason for that is, for a long time, I had wanted to know what these proteins do in human skin. We knew they were increased in scleroderma. You can do all the testing you want in the lab. You can do all the testing you want in mice. That doesn't mean something will ultimately improve the treatment, because many, many of the drugs that are tested in animals in the laboratory or in cells in the laboratory fail when they get to the clinical trials in humans, because we are not mice. We're not going to respond the same way as mice. So, and I had always wanted to just inject a human skin. Can't find volunteers for that one when you're working with unknown proteins. Not that you could get approval to do that one at most academic institutions. So I saw that quote, and I made up my mind that when I came back from vacation, I was going to find me some human skin. And that's what I did. And this is what it looked like. I'm so glad you guys had dessert right before this. This is human skin from plastic surgery. This is from paniculectomy, which is tummy tuck, basically. And you get the skin, and believe it or not, that thin layer up here is the skin. This layer, that's all fat. Yeah, it makes you regret that dessert, doesn't it? So basically, this is the fat. You trim off the fat. You just kind of peel it off, pretty much. And we used the skin, and we thought we're going to take these proteins we discovered, and we're going to inject them in human skin. I think a lot of people I know thought I was a complete lunatic at that point, but so we cut it up into pieces. We put it in the lab in these plastic dishes that we use. We let the upper part of the skin, known as the epidermis, stay exposed to the air, because normally that's how it is. And we put this nutritious medium that we use in research around the skin to keep it alive for a week or two, so we could actually grow the skin in the lab for a week or two. And then we injected it with a variety of things. And the injections are easy. If you've ever had a TB test and you get that little blister, it's pretty much that way. And then we waited a week, and we waited 2 weeks, and then we collected the skin to see what happened to it. And if I were to tell you that the proteins we discovered when we put them in human skin caused fibrosis, just looking at this picture, I think you would believe me, because look how much thicker this piece is, just to the naked eye. But if you look at it under the microscope, so this is if you take the skin and you just slice down into it, so this would be the upper part of the skin, the epidermis, that's exposed to the air, and then as you go deeper into the skin, this is what normal skin looks like. This is what skin where we injected the proteins that were increased in scleroderma looked like. So you can appreciate how much thicker the skin got. And that told us that these proteins can trigger the fibrosis. They can actually initiate it on their own when you inject it in skin. But it also told us that this works in human skin. And if we can find a therapy that reverses what we see here in a human skin, then we'll have a higher likelihood of succeeding when we get to the human clinical trials. Make sense? All right. So we started with an observation that these proteins are high in the organs of scleroderma patients. We also looked at other diseases like idiopathic pulmonary fibrosis, and they were high there, which told us that this link may be useful for other diseases that have fibrosis. We checked them on cells in the lab. We checked them in mice, injecting them into mouse skin, into mouse lung. And ultimately, we took them back to the human skin and showed that they can cause the fibrosis in skin. So great, we can cause fibrosis. But we need to understand how. Because if we understand how, we can stop it. And the trick to understanding how is this molecule. And you can look at it and say, but it's a collagen. Yes, we know that we make more collagen than we should. And that was my first thought. We made this observation, and we let it sit, because we realized that these proteins we're injecting that cause fibrosis are increasing this collagen. And I thought, okay, another collagen. We know they're causing fibrosis. It makes sense they increase collagen. But there was a caveat to this collagen, in that this end piece of this collagen gets cleaved off. It gets chopped off, and it's called endostatin. And when I read up about it, I realized that this molecule had been undergoing now phase 4 clinical trials in cancer to prevent metastases in cancer, because it stops new blood vessels from forming, which stops the cancer from spreading in the body. So I thought, okay, so obviously collagen has other functions other than just making the tissues thicker. So we decided to look at it. So we knew our proteins called IGF-BPs increased this endostatin, basically this piece of collagen. But the question was, could then endostatin by itself cause the fibrosis as well? So this is the sequence of things, how they happen. So what we did is we took now, just like we had taken this protein and added it to cells to turn them into scleroderma cells, now we took this endostatin and we edited to cells in the lab, and then we looked. And what I'm going to show you, I know you've never seen data from a lab, but it'll be easy to understand. If you see these dark blobs, the thicker and darker the blob, the more there is of a protein. So this is showing collagen. So this is fairly thick. There's a lot of protein there. But what you can see is everywhere there is the letter E, that's where we added endostatin. And I think without being scientists, you can appreciate that each time we add endostatin, we really reduce the collagen levels. They're almost gone at this point. And we reduce levels of other, what we call extracellular matrix proteins, other proteins that are made in excess with collagen and that bind collagen and contribute to the organs becoming thick. And to us, of course, that was the wow moment. Well, this is really me with the curly hair. That was the wow moment. Because we were expecting that this endostatin was going to do what the protein that increases it does, which is make more fibrosis. And we looked at it, and what we saw is a shutdown of fibrosis and a reversal of fibrosis. And I can tell you, I did not sleep a wink that night because I knew exactly what we had. And this is where this quote from Louis Pasteur, it's my favorite quote, "Chance only favors the prepared mind." We were expecting the experiment to be the reverse of what we had. But we had an open mind and accepted that the answer was opposite of what we expected. And that's the reason we are where we are. So we did the typical experiments. We tested it in mice. And the reason we tested again in mice, we wanted to cause the fibrosis. And then we wanted to wait a week and then add the endostatin peptide that we had, because we wanted to know, will you make a difference if the fibrosis is already there? Because when you get to your doctor, you already have fibrosis. So being able to prevent it is not as useful as being able to reverse it. And basically, all this is going to show you is this is skin, again, but this is mouse skin at a cross-sectional, and you can see that our peptide really reduces the fibrosis. So we're comparing it to an agent here that causes fibrosis. I don't want to show you a lot of data. I just want to show you what we do. And the way we cause fibrosis in mice, pay attention, there's a quiz. We can inject growth factors that make the mouse skin thicker. Then we add our peptide and see if we can change that. We can inject something that's a chemotherapy drug used for cancer called bleomycin. And that's because patients who were receiving this chemotherapy started developing lung fibrosis. And then doctors realized that the bleomycin, which was supposed to cure their cancer, was causing the fibrosis in their lungs. So we took advantage of that observation, and we injected into mice to be able to make a model of fibrosis. So obviously, we were happy with that. And then we went back to our human skin. So now it's human skin to make sure we could get it to work. And obviously, looking at this, compared to how thick this is, it does work. And even if you wait and give it after the fibrosis in the skin, it still works. So we were very excited. So the key to not just stopping fibrosis, but to be able to reverse it and bring the organs back to normal is what we were missing. And we needed to understand how this peptide was working to do that. And we finally understand how it works, at least one mechanism. And the key was in the matrix. What I mentioned is the extracellular matrix. And when I say the matrix, most of you will think of the movie. But that's not really what we're talking about. What we're talking about is this. This is a cell, highly magnified, and this is a collagen molecule. So you can see collagens, there's other molecules around them that are produced that are called extracellular matrix proteins, and they start interacting together like here, and they cross-link. And the more they cross-link and link each other, the stronger the matrix is and the stronger the tissue is. So basically, if you have this as collagen and you have a lot of cross-linking between the collagen molecules, the tissue is stronger and more fibrotic. If you can reduce the number of cross-links, the tissue becomes weaker, and then it's easy to break it down and reverse the fibrosis. And it turns out that's what we were doing. That's what our peptide was doing, even though at the time we didn't know it. But basically, our peptide was reducing these cross-links, the number of them between the collagen molecules. And then these enzymes, like Pac-Man, that come and chew the collagen, could access the collagen, because now the collagen was looser. It wasn't as tightly bound. And it could access the collagen, and it could break it down. And that's the key to be able to reverse the fibrosis. So where are we? A patent was issued that remained at my previous institution, the University of Pittsburgh, and a small company called iBio licensed it from the University of Pittsburgh. The key to iBio is they have this unique technology in that they're going to make the peptide, they already are actually making the peptide in plants. And you can just chop up the leaves of the plants, extract the peptide, use it for a therapy. And we're hoping to get to phase 1 clinical trials in 2015, which is not that far away. And our goal is to possibly start phase 1 clinical trials instead of starting with healthy volunteers, actually start with patients to know right away how effective this will be. And if it is effective, I would urge you to keep a try on the right to try law that's being discussed in a lot of states that will give faster access to all patients to a drug before it's actually available. So this is where we are with our story with the peptide. And as you see, it's very much an accidental discovery. It was accidental. We were looking at something that causes fibrosis to see how it causes it, and we stumbled on something that had the opposite effect. And a lot of discoveries and research are accidental. Many of the things we use, antibiotics, antidepressants, anesthetics, even surgeries, types of surgeries, transplants of organs, a lot of them were the result of accidental discoveries in the laboratory. Let me give you some examples. The microwave. So the way the microwave was discovered very accidentally. So this gentleman by the name of Percy Spencer was working in a company that had a magnetron. And a magnetron is like a vacuum tube that can be used to generate microwaves. This was 1945. He walks in front of the magnetron, and he had a chocolate bar in his pocket, and it melted. So he decided he would look into it. And he tried a variety of things, including one experiment that involved the explosion of an egg. And pretty soon after that, he came up with the first microwave. So accidental, walking in front of it, chocolate melted, he put 2 and 2 together and came up with a microwave. Here's an accidental discovery. I don't know how many of you know that story. No one recognizes this drug, I'm sure, but Viagra was accidental. So Viagra was actually developed and used in clinical trials, and the intent was for it to help alleviate chest pain in people who are having chest pain. There were a lot of men enrolled in that study. The clinical trial failed. Viagra did not work, did not improve chest pain. And when they tried to ask all the volunteers in the clinical trial to return the unused drug, they couldn't get any of it back. And when they looked into why they couldn't get it back, they realized it had a very beneficial side effect. Ended up being a blockbuster drug for something that it wasn't originally intended for. All right. Here's another one, penicillin, an amazing antibiotic. The way this was discovered was by a Scottish biologist. Fleming is the name. And Alexander Fleming used to work on bacteria. And this, on this side here, is a petri dish. And these are colonies of bacteria. He worked on staph. Many of you have heard of staph. So he worked on these bacteria. And he went off on vacation in August, typical European holiday time. And he forgot his petri dishes on the bench, sitting outside. He forgot to put them in the fridge. Came back and noticed that a fungus grew on his plates that sat on the bench while he was on vacation. But at the same time, he noticed, and there's where chance favors the prepared mind, that all the bacteria that were growing here around the fungus had died. So he realized that this fungus was making something that kills the bacteria, which led to the discovery of penicillin. Another potent antibiotic, Magainins. So this was discovered by Michael Zasloff. He was doing surgeries on frogs. He would do surgeries to remove the ovaries and stitch up the frogs, and then toss them back in the aquarium in the tank he had in the lab. And the tank had really murky water with a lot of bacteria. It was nasty water, and he realized that the frogs were healing the incision very quickly and were not getting infections in spite of the fact that the water was very dirty. So he pursued it further, his observation, and realized that the skin of the frog was making these peptides that we call Magainins, that are very strong antibiotics. All right, a few more. Insulin. Most of you have heard of insulin for diabetes. So insulin was discovered when these German scientists were doing experiments on dogs. And I don't like the idea of dogs, so that one we won't get excited about. But they were taking out the pancreas of the dogs surgically because they wanted to understand what does the pancreas do. At the time, they didn't know. And one day, one of the lab assistants comes and gets the scientists and tells them they have to come and see something. And they come in, and the dogs that had the pancreas removed had a lot of flies swarming around their urine in the cages. And so they looked into it further and realized the urine, when they tested it, had a lot of sugar in it. And that's how they made the association that the pancreas must be making something, which is insulin, that controls sugar. And when it's defective or insulin is not there because they had taken out the pancreas, a lot of sugar is produced. So that was the first example of experimental diabetes. Infant jaundice. So many of you who had preemies as babies know that they often get jaundice. And one of the first things they tell you to do is put them near a window, and the jaundice goes away. And that's because in the 1950s in the UK, a nurse made the observation that all the babies in the nursery had jaundice who happened to be closer to a window and got some sunlight, had the jaundice resolve. And that made the connection that the nearly transparent skin of infants, newborns, can take up the UV light from the outside light to process and break down the bilirubin that causes the jaundice and cause it to be excreted and resolve the jaundice. Again, another observation that's now still applied to babies everywhere. All right, let's go into sugar. Sweeteners, artificial sweeteners. We'll start with the first one. The first artificial sweetener to be discovered is saccharin. It was discovered in the late 1800s by Fahlberg while working in the laboratory. And the way he discovered it, they were doing chemistry, and he spilled something on the bench. Went home, had dinner, was eating bread, and realized that the bread tasted awfully sweet. He had not washed his hands. And whatever he spilled in the lab was still on his fingers, tasted sweet, and led to the discovery of saccharin. Bad habit in the lab. We don't do that. We wash hands. All right. Aspartame, another sweetener. This was discovered by someone else, another chemist, who was working on treatments for gastric ulcer. And it's interesting, because what he did is he spilled again. You know, for some reason, the substitute sugar stories are all sloppy chemists, but he spilled something on his bench, didn't wash his hands, and did what a lot of people do without thinking, licked his finger to flip a piece of paper, and then went, that tasted awfully sweet. Proceeded to taste everything he had spilled on the bench in the lab until, bad habits, until he found the sweet spot, and that's how aspartame was discovered. All right, another sweetener, cyclamate, that was also discovered. This one was by a student in the lab. Again, accidental. What do you think he did? No, this one didn't lick his fingers. This one was smoking in the lab, another no-no. Set the cigarette down on the bench, on the edge of the bench, to do his finished part of what he was doing, then picked it up, put it in his mouth, and tasted something sweet. Proceeded to check which one it is, and found this one. And the very last one is sucralose. And that one was discovered because the student in the lab was a foreign student who didn't really understand the language well. And his supervisor told him to test the chlorinated sugar. And instead, he understood he had to taste the chlorinated sugar. Proceeded to taste all the chlorinated sugars and found one that was really sweet that ended up being used as a substitute sugar. So again, very accidental, very sloppy, but very accidental discoveries. All right, two of our most important modern drugs were discovered in sewage and dirt, believe it or not. In sewage, in Sardinia, they isolated cephalosporin, which is an antibiotic, that was in the water where the sewage was being pumped out in the sea. And that's how they identified it. And the other one was in dirt. And this is by someone who worked for a company that used to send people out to different countries to bring back soil, and then they would test the soil for the presence of natural antibiotics. And basically, he had brought back some soil from Norway, and they tested it, and it didn't have any antibiotic effect, but they found that it really suppressed the immune system, and that's how cyclosporin was discovered. All right, last example is a more recent one. Actually, this one I read about, I want to say maybe 3 months ago. So these investigators at Johns Hopkins University were doing experiments about cancer in mice. They would take these mice, inject cancer cells in them, and the cells grow into tumors in the mice. And that's how you study cancer in mice, and then you can test different things in the mice. Well, they got a batch of mice where they tried to repeat their experiment, inject the tumor cells, but the tumors wouldn't grow. They got another batch of mice, tried to inject again. They couldn't figure out what they were doing wrong. Then they realized that there had been an outbreak of pinworm in the animal facility where they were doing the research, and the mice were being treated with pinworm medication. Turns out this pinworm medication prevents the growth of cancer, and that has led to a planning of a clinical trial to test this medication in patients with brain tumors. So stay tuned to that one. Again, accidental discovery. So this is the research we do and the discoveries we do, but what can you do? You actually have a role in research, and you have a critical role in research. Patients play an important role in getting funding for their disease, but also in making the therapies available. And I'm going to give you real-life examples of how that happens. Gleevec. Many of you have heard of Gleevec. It was introduced for leukemia, but the company wasn't really interested in investing a lot in it because this form of leukemia, CML, was not that common. So this was not going to be a big market for them. But when they started the clinical trials with patients with this leukemia, it was very effective. Gleevec was very effective for this particular form of leukemia. So the patients got involved before and during the clinical trial. They put together a major effort, and it's because of their activism that there was a record approval of Gleevec through the FDA to make it available to the patients. So that tells you you have the power to impact what happens in research and what results from our research. Here are other examples, 3 of them. The National Multiple Sclerosis Society pushed through the National Multiple Sclerosis Act in 1949, which led to increased medical research funding on MS. MS has quite a bit of funding at the National Institutes of Health compared to scleroderma, considering the numbers of patients with each. ACT UP, which is an organization to support patients with HIV and efforts to eradicate HIV, through their efforts in the 1980s, were able to increase the NIH budget for HIV research from 21 million to 741 million, boosting research on HIV. The Breast Cancer Association, which was formed in 1990, was able to push the federal government to increase research for breast cancer research from 90 million to 800 million. So this is the type of effort you can do with the advocacy that the foundation is involved in, with keeping an eye on what's happening with clinical trials and voicing your needs and your views to the FDA, to the federal government, to the National Institutes of Health, to your congressman, to your representative. You can actually make a difference. All right, the Scleroderma Foundation has been involved in funding research in scleroderma. I know many of you are aware. I'm just linking how we do research from how it starts from you. So when funds are donated for research, research grants are submitted by scientists like myself. There's always a deadline for the Scleroderma Foundation. It's September 15th, and all these grants come in on September 15th. The grants get sorted through, and then they get assigned to scientific experts who are going to evaluate the quality of the science in them, because the Scleroderma Foundation has a peer review system, which means that grants are scored based on their scientific merit, not based on who submits them. We want to fund the best science. Then in November or December, the scientific experts meet face-to-face so they can discuss the grants face-to-face. They score the grants based on scientific merit, and the grants that receive the best scores are funded, and the recipients get the announcement. So this is how it currently works. We have 2 types of grants: the established investigator grant, which is given to people with more expertise and experience in research, and the new investigator grant, and the goal of that is to get people started in research and support them, because when you're starting off, it's really hard. There's not a lot of money for research. You don't know a lot. You need to have mentors. You need to work really hard. So we want to make sure that there's a next generation of researchers. I hope what we do ends up putting us out of business, but there's always going to be a disease that's going to need scientists to work on. And to date, more than $20 million have been committed by the Scleroderma Foundation to research. The Scleroderma Foundation is actually the largest funder of scleroderma research after the National Institutes of Health, which is an incredible accomplishment. We have started a brand new grant initiative, and the goal of it is to encourage and promote collaboration across different scleroderma centers because they can complement each other's expertise and accelerate the pace of research if we all work together across centers. So it's called the Multi-Center Grant, and the first one will be given this year, and it's to the tune of $500,000. So that's a new grant opportunity. So that gives you an idea what the Scleroderma Foundation is doing in addition to conferences, patient support, education, awareness, advocacy efforts. This is another level of support that they're providing to the researchers to advance research on scleroderma. So I know that seems like a lot of money to you, but in the big scheme of things with how expensive research is, it really isn't. And I love this quote by Mary Lasker, who said, "If you think research is expensive, try disease." All right, so I hope from what I told you today, you'll realize that we're working hard to solve the scleroderma puzzle. We're working hard in developing therapies, and finding a cure is not a mission impossible. I'm confident we will get there. The question is when. I often get asked by potential donors, well, how much money will it take to find a cure? How many $1 million? And I think you can appreciate from what I talked about, the accidental discoveries, that it could be millions of dollars, or it could be the next dollar spent in the lab with the next observation we make. We really don't know. Thank you for your attention. So for the sake of time, and in order not to keep you here late, I'm happy to stay here and answer questions, but for those of you who might not want to stay as long, here's the quiz. All right, you guys ready? Yeah. We're going to play Jeopardy. Let's see how well you paid attention. And Robert mentioned what an amazing staff the Scleroderma Foundation has, and I echo that and add to it. We have the most incredible staff of any organization in the world, and I know that for a fact. And they've been working really hard for this conference, and they're here. So you'll have Anne, Ryan, Maureen, Shenna, Christina, Carrie somewhere, and Robert's going to pitch in too. And what they're going to do is going to have microphones ready for you. All right, we're going to start over there first. So anybody from that first section can answer the question. And here are the categories. And I'm going to put Robert on the spot and ask him to pick a dollar amount and a category for that first section.
Gee, Carol, I'll take models for 200.
Models for 200? This chemotherapy drug, remember to raise the sheet of paper if you want to answer. This chemotherapy drug is used to cause fibrosis in mice.
Bleomycin.
Remember Jeopardy, you have to answer in a question format.
What is bleomycin?
She said, what is bleomycin?
Good, you got it.
Okay, I'll take this and that for 300.
This and that for 300. This nonprofit organization funds scleroderma research. This section. I think Joey raised first.
The Scleroderma Foundation.
Question?
Scleroderma Foundation.
What is the Scleroderma Foundation?
Works. Good job.
Take a chance for 500.
Take a chance, 500. A drug used to treat this parasite may be effective for brain cancer. What is pinworm? What is it? Perfect. And I'm going to ask you to pick the next category, please. Ma'am, please pick the next one.
Kara, discoveries for 300.
Magainins are antibiotics discovered in the skin of this amphibian.
What is a frog?
Yes, sir. What is a frog? And you get to pick the next category, please.
I'll have fibrosis for 400.
You got it. Fibrosis results from increased production, the first section, increased production of this protein.
What is collagen? What is collagen?
Correct.
Models for 400?
Models for 400? For this section now, right here. Before testing a potential treatment in humans, we must test it in this animal. Wow.
Right there, right next to shut up.
What is mice?
Correct. What is a mouse or what are mice? Absolutely. All right. Would you please pick the next one?
Take a chance for 400.
Take a chance 400. This section. Your turn. Okay? This is the dollar amount given by the Scleroderma Foundation for research grants. I'm sorry?
$2 million. What is $2 million?
No, ma'am. I think the lady in the orange topping, I can't tell.
Okay. What is 1.5 million? Annually.
Back.
What's 20 million?
You got it. What is $20 million? And I will ask you to pick the next one, please, for the last section there.
I'll take discoveries for 500, please. The next section. Fine.
You got it. All right, last section. Insulin was discovered when flies swarmed the urine of this animal.
What is the dog?
Correct. Yes, sir.
This and that for 500.
This and that for 500. You guys ready there? This person, so typo, said, if you think research is expensive, try disease.
Who is Mary Lasker?
Question.
That's what I said. Who is Mary Lasker?
Correct. Thank you. And the next category?
Oh, this and that for 400.
This and that 400 for this section. This government agency funds scleroderma research.
What is NIH?
Correct. Thank you.
Okay. Take a chance for 300.
Take a chance for 300. This section. Ready? Cephalosporin was discovered in this.
What was sewage?
You guys were really paying attention. I'm impressed. All right, next category for that section, please.
Oh, yes, and then, sorry. Models for 500.
Aiming high. Let's go. Many discoveries in science and medicine occurred in this way.
What is by accident?
Correct. Or accidentally.
Fibrosis for 300.
Fibrosis for 300. You guys ready over there? Here we go. To prove something causes scleroderma in mice, we check these organs. Uh-oh.
What is skin?
That's one correct answer. You had options.
Okay, next category. Okay, next category. For $400, discoveries.
For discoveries for $400, this is going to be for this section. Okay. This person discovered the microwave when chocolate melted in his pocket. He loved chocolate. That's a good thing.
Who is Spencer?
I didn't hear. I'm sorry.
Spencer?
Question?
Who is Spencer?
Correct. Percy Spencer, yes. Close enough. All right. And we'll let you pick the next category to be answered by this section.
Models for 300.
Models for 300. Everybody here ready? Here goes. This tissue is discarded from plastic surgery and can be used to study scleroderma. This tissue.
Skin.
I'm sorry?
What is a tummy tuck skin? Skin from tummy
What is
tuck.
skin? That's good. It could be other tuck too, but.
This and that, or taking chance for 500.
Take a chance, no, we did 500. 200? Okay, this is for the section over there. Take a chance for 200. This is the first substitute sugar that was discovered. We need a microphone.
What is saccharin?
Correct.
Fibrosis for 500.
Fibrosis can be described using this lay term.
What is thickening?
Correct.
Oh, sure. Let's see what we have. How about discoveries for 200, please?
Discoveries for 200. Now this section, you're on. This person discovered penicillin.
Louis Pasteur.
No. Up for grabs, guys. Raise the.
Who is Fleming?
Correct. Alexander Fleming. All right, next category.
Let's do fibrosis 100.
Fibrosis for 100? Okay. This section, you're on. Fibrosis for 100. Fibrosis and scleroderma can affect this organ.
What are the lungs?
Correct. That was one option. You had a lot of options.
I didn't even think of any other organ. Oh, how about this and that for 200?
This and that for 200. Section over there. This antibiotic was discovered when a fungus grew on a plate.
Penicillin?
Question?
What is penicillin?
Correct.
Models for 100?
Models for 100. Okay, we're going back all the way over there. You guys ready? All right. This animal is commonly used to study scleroderma.
What is a mouse?
What is? I'm sorry?
A mouse.
Correct.
There you are. In the next category?
Discoveries for 100.
Discoveries for 100, and this will be for this section. An example of an accidental discovery for jaundiced infants is this.
Was that eliminating the jaundice because of the infants by the windows?
So what is your answer?
What is the eliminating of the jaundice? Oh, what is sunlight? Thank you to my wife.
Okay. 3 more to go. We'll let you pick the category for us for this section to answer.
What is fibrosis for 200, Alex?
Fibrosis for 200. You guys ready here? To measure severity, and I didn't go over this, but a lot of you have it, of scleroderma, your doctor pinches this.
What is your skin?
Correct. 2 more.
Oh, take a chance for $100.
Okay, this is for that section. This person said, chance favors the prepared mind.
Pasteur? What is Pasteur?
Who is Pasteur?
Correct. Louis Pasteur. All right, and the last question is going to go back to that area. You guys have been so patient. I hope you're enjoying this. All right, ready? This and that for 100. This drug for erectile dysfunction, ED, was discovered while treating patients with chest pain. Everybody can answer that one.
Viagra? Oh, what is Viagra?
I'm sorry, I didn't hear.
What is Viagra?
Correct. What is Viagra? Correct. Nice job. Now, I noticed one thing. We have 3 young players that I could see who tried very hard, but they had a height disadvantage. They did raise the paper, but we missed them. They're sitting in the front right there, and I think just for the effort, they're going to get prizes. Do we have any other young players that I couldn't see from up here? There's one right there who lifted a paper too. I think she's earned a prize. Did I miss anybody else back there? Maureen, you got, okay, and over there. Anyone else who has a height disadvantage is under the age of 15? I would qualify if there was no age restriction. Anyone else I missed who's under the age of 15? Doesn't help to shrink down. No, they're joking. All right, you guys have been a tremendous audience. I'm happy to answer any questions. I wish you a great conference and a great weekend, and thank you for letting me be part of this.